today I'm going to be presenting my results and what we did from day 6 to day 10 basically. Sorry, the slides are. Okay. So first of all, on day 6, basically we didn't do much, but we actually went on a very educational trip to Biotechnology, which is a forest in UM basically, and we actually learned a lot of interesting things. So I actually took a few photos of basically our trip, and here's a few facts that I learned basically. So when we first entered, we learned that one of the leaves are actually it used to be used for writing, and it's something that's on the UM logo. And also we learned that there's a vine that crawls up the trees, and that actually functions because they cannot grow themselves. So it's kind of basically the ecosystem helping each other, but it's also kind of basically plants competing to get the most resources, which is sunlight and nutrients obviously. And also we saw the biggest leaf in the world, which is something I didn't know existed. I mean, I kind of expected that it existed, but I didn't expect that it was in Biotechnology. And also we saw a class A water, which is something that's really clear and clean. So yeah, that's basically just our trip to Biotechnology, and here's more of the photos. Okay, so basically on the next day we basically did microbial analysis. So we learned about the importance of measuring the levels of microorganisms in food samples, and also I'm also going to be mentioning the methods and also the results that we got. Okay, so why do we need to do microbial analysis in the first place? So I think it's pretty common sense, but basically we test for microbes because microbes are everywhere, and we need to test for the presence of yeast and many other microorganisms. And this is especially important, especially in the food industry, because we need to measure food safety and also the standard of consumption. Okay, so how did we do this exactly? How did we actually analyze? How did we actually do analysis on the food samples? So basically we used a Petri film, which is coated with agar gel and nutrients, which will help the microorganisms grow. So when we incubate it, you can actually see it visually. And considering that we had many different types with the aerobic count, they're also colored differently so that it's easier to visualize the different types of microorganisms. So basically we started off with the distilled water as and then we also loaded the food samples onto the Petri film. So you might also be familiar with this, because this is also something that you see with agar plates. But something that I found out is that these Petri films are actually they're similar, but they're cheaper and they're easier to use. So basically the method we did is to simply pipette the sample onto the Petri film and using a spreader to evenly distribute the sample. So basically after doing all this, which doesn't take a really long time, you leave the sample to solidify in the gel, and then you basically you incubate it in a water bath to make sure that the microorganisms grow. And a very important thing that we had to make sure of in this experiment basically was to make sure that it was as hygienic and sterile as possible. If not, the possible contaminations could possibly interfere with your results, such as your spit or your fluids or whatever. Okay, so basically these are the results that we got. So a bit of an interesting anomaly that we got was that our control, as she said earlier, had some growth in it, and that was a bit weird because it's not supposed to have any growth, because distilled water is supposed to be pure, right? But we thought it's probably because there was some contamination going on. But it was also interesting to note that the lemon also did not have any growth at all, which is likely a result of the fact that it has antimicrobial properties in it. And also you can see the results from ripe mangoes, and we did multiple tests for each fruit. So this is basically the control. This is unripe banana, and they each have a sort of similar results, but you can see that they really differ. And the way you can actually count this is by basically counting each grid, and which is kind of the reason why it's there in the first place, which makes it easier to count. Okay, so the next thing that we did is protein extraction and quantification. So the reason why it's important to do is because protein is a vital nutrient, and companies need to know if their foods have high protein content or not. So our bodies need protein for building tissue and supporting overall health and stuff. Okay, so how exactly did we do this, and how exactly did we actually measure the protein content? So basically we used the fruit extract, and we put the dye inside, and we did a lot of centrifuge and stuff. and then basically we actually put this inside like the glass like tubes and put it in a spectrophotometer, which will actually measure the total protein content. So basically we did this. After we did this, we incubated the mixture, and then we put it in the spectrophotometer at like the exact like nanometer of like 595, and then you can actually input it into a formula, which is to calculate the total protein content. And these are the results that I got, and I don't know how to put it in a table actually, but you can see that actually like, what is it, unripe banana had like the highest like overall, and like the results are actually down here, like not up here actually. Okay, so I'll just move on. Okay, so now lastly, the last and final experiment that we did was separating the protein using electrophoresis, which is like an SDS page basically. So SDS page stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis. So this is actually used to basically like denature and coat to ensure that all the proteins are negatively charged, because you are using an electric current and you want to make sure that they're all uniformly negative. Okay, so then an electric current is basically run through to separate the protein based on its weight, and the heavier proteins actually stay at the top, while the lighter ones can pass through the pores. So you can see like a clear separation based on the weight of these proteins. So then there is after you finish everything, they can actually be sent for sequencing, where based on the patterns of the results that you got, they can actually be different proteins can be identified. So this is sort of like a diagram of kind of like explaining how we did it. So basically we used the different samples of, we used different samples of fruit, and we actually pipetted it into two different like sections like of gel. So you can see that there are different wells, and they will pass through the resolving gel. And based on the resolving gel, it will be like filtered through, so you can actually easily see the weight of like each protein and measure and sequence it after that. Okay, so basically this is like the result that we got. So like I basically made it big so you can see. So basically what I found out is that they actually give you a base protein, which is like something that you can use as like a measurement, as a sample to compare it with other proteins. And you can see that the heavier ones will definitely like move to the bottom. No, the heavier ones will stay at the top, while the lighter ones move to the bottom because they can seep through. And I didn't label it, but an interesting thing we learned is that with a certain type of banana, there's a certain protein which basically prevents like the pests from eating it in the first place. So yeah, these are the results that we got. Okay, so like that's it. Thank you.
Thank you, Mariam, for that presentation. Just comment on the gel picture. It's upside down, so it's not the true representation. So you must take some care about presenting the gel in the correct form.
Oh, okay.
Just one question, based on week 1 and week 2, what do you think is the major difference between agarose gel electrophoresis and SDS-free electrophoresis?
So I think in terms of methodology, they're similar, right? But we know that one is used for separating DNA and one is used for separating like proteins. So we know that they're different because like obviously like other than the basic methodology, I think that like proteins is separated like based on like the weight of it and how it travels through like the pores, and the DNA separates like differently than the protein does.